The overall goal of this procedure is to rapidly identify and test possible domain boundaries within large coiled-coil proteins to improve the likelihood of finding an intrinsic fragment that will crystallize.
This method can help researchers streamline the process of crystallization for large coiled-coil proteins, which can be notoriously difficult to crystallize. The advantage of this technique is that it integrates both experimental and computational methods to quickly identify and screen likely candidates for crystallization, thereby improving their chances for success. Demonstrating this technique will be Jenna Zalewski, a graduate student in my lab, and Keith O'Connor, an undergraduate researcher in my lab.
To begin, combine output from established web-based tools to generate predictions of possible coiled-coil domain boundaries, as described in the text protocol. After generating expression plasmids and testing their expression levels as detailed in the text, begin purification of each strain that successfully expressed ruby-tagged protein. To lyse the cells, first resuspend each thawed cell pellet in 1.5 milliliters of lysis buffer that has been supplemented with protease inhibitors. Add 30 microliters of lysozyme at a concentration of 10 milligrams per milliliter and incubate at room temperature for 20 minutes. Then, sonicate following the instructions for the instrument being used. Centrifuge the sample in a tabletop centrifuge for 30 minutes at 14,000 times G to pellet insoluble material or unlysed cells. Save both the supernatant and the pellet for subsequent analysis via SDS page. To perform nickel affinity purification,